Hi guys, Mrs. H here. Today we're going to be looking at the cell cycle and mitosis. So we're going to be describing what happens during the cell cycle and then what happens during mitosis. Here is our key vocabulary for this lesson. so, as you know, all cells start out as a single cell, whether you're a single cellular organism or a multicellular organism. And so, in order for a multicellular organism to uh, grow and develop into a bigger multicellular organism, a cell cycle has to occur, including mitosis. Now, if I'm a single cellular organism, I will also go through the cell cycle, but the difference is at the completion of the cell division, I will actually have two new individual organisms. So let's take a look at what's actually occurring during the cell cycle. So, when we're looking at the cell cycle, we're actually looking at three things occurring. We're looking at growth, DNA replication, and cell division. So, we've already looked at in detail DNA replication. So, now we're putting these pieces of the puzzle together. So, a cell will grow. And yes, cells do grow even if it's one singular cell. They do grow. They will undergo DNA replication, and when conditions are favorable, the cell will divide. And after that cell division, we will have two new identical daughter cells. These cells will be clones. And so let's look at this cell cycle. So there's two major parts of the cell cycle: interphase and mitosis. Most of the time, the cell will be in interphase, about 90%, give or take, depending on the cell. Now, each cell, depending on the type of cell, that time period will vary. So, bacteria, they reproduce very quickly, but still, if you look at from one cell division to another cell division, 90% of the time is in the interphase. Now, looking at other cells and its rate of replication, they may be spending, hours and even days in that interface. So that is variable. And let's look into what's happening at each one of those phases. So interphase is broken into what we call from cell division to cell division. And so in between each cell division is actually the interphase. And so this cell will grow in size, it will replicate its DNA. And the reason why we need to replicate its DNA is because the new daughter cell needs its own DNA in order to survive and function. And so that interphase is when the DNA replication will occur and when growing will occur. And so oftentimes this is considered the resting phase, even though it's not resting. Cells are very active. They're producing enzymes. They're making those building blocks for the DNA. They're performing cellular respiration. If they're a plant, they're undergoing photosynthesis. So even though it's considered um, a resting stage, it's really not. The cell is very active. And so if we're looking over here at this diagram, this almost the entire um, cell cell cycle. So if this is start and this is the end, most of the time is spent in that interphase period. So now interphase can be broken down into subphases or sub groups or sub time periods. They're called G1, S, and G2. Um, G1 is the cell growth phase 1 or first gap. S phase is for synthesis or DNA synthesis, which means to make. And then gap 2, um, second gap, which is G2, which is post synthesis. And so each one of these phases has very distinctive things that are occurring during these time periods. So if we're looking at the first gap or G1, this is when even in terms of the cell, this is usually when we're looking at a cell. What we think of when we see a cell is what we're really looking at is that G1 phase. Um, there's really not much to see in terms of if you're looking at under a microscope. However, that doesn't mean there aren't a lot of activity going on um, in this. So, um, like I said before, this is a high energy, lots of activity, lots of things going on. This would be like your routine day to day function. So, as a cell, so whatever that cell is doing, whatever the function of the cell is, whether it's a brain cell or a liver cell or a stomach cell, this is it. And this is, I'm being personifying cells and talking about humans. Um, but this is for any organism, any multicellular organism, and even for a bacteria. This cell is performing its day to day function. This is routine work going on during this time period. So then we have S phase. So this is when DNA replication occurs. So this is when the cell is taking its DNA, it's using those enzymes, and it's making an exact copy of itself. This is when we've got the topoisomerase, we've got the helicase, we've got the polymerase, we've got the ligase, we've got all that activity going on the leading strand and the lagging strand. This is all what we had previously learned in the chapters before. So if we look at it in a picture, this should look pretty familiar. We have our original DNA strand. So we get the enzymes involved that's splitting it open, unzipping it, breaking those hydrogen bonds, holding it open. And then we have the polymerase coming in and adding those nucleotides on the leading strand. It's running through continuously. Lagging strand, it's making those Okazaki fragments. So you should be familiar with what's going on here. This all occurs in the S phase. That's what's happening in S phase, is that DNA replication. So what happens after DNA replication occurs? This is when um, so in humans specifically, this is when what we say is our chromosome numbers double. So for humans, we have 46 chromosomes. So after S phase, we have 92. So that each new cell has its own copy of chromosomes. Now, if I'm a fruit fly, I'm not going to end up with 92 chromosomes because I don't have 92 chromosomes. I'm a fruit fly, so I start with 8. So if I double my chromosome number, I'm going to end up with 16. So it's variable depending on the organism that is replicating and having its DNA replicated. So, what's interesting to note here, and this is something new that we hadn't talked about, is what actually happens to the DNA. So, the DNA will actually replicate itself, but the two new strands don't separate completely independent of one another. They're actually going to stay kind of stuck together or stapled together. And that staple, if you would, is called a centromere. And so centromeres hold two identical strands of chromosomes together. Those two identical strands of chromosomes are called sister chromatids. So when I go through DNA replication, I have my original strand and I have my new strand. These are identical. There's nothing different from them. They're it, they are going to stick together with a centromere. So they're going to buddy up and stick together. So if I have chromosome number 1 copied itself, chromosome number 1 sticks together by a centromere. And that's going to play a part when we get into mitosis. So then what happens? Well, and to note here, and I didn't mention this earlier, a cell will not replicate its DNA unless it's healthy and the cell is ready to divide. It will stay in that growth 1 phase until a checkpoint. And basically, a checkpoint is it a cell's check is its balance, is its check and balance. So if the cell is healthy and everything is good, it will say initiate DNA replication. It will initiate that S phase. So if there's anything wrong or the cell isn't healthy or there's abnormalities, it will not pass through that checkpoint to go into DNA replication. So now we have DNA replication. Now we're going into G2. And so now what's happening here is it's a growth period, but this growth period is getting ready to produce 2 new cells. So it's stocking up on everything so that it has enough energy, it has enough organelles, it has enough supplies to sustain 2 cells. So imagine, if you would, that you are living in one house and you start stockpiling all your material so that when you're going to go buy another house, so that your house 1 and your house 2 has everything that it needs for you to live in both houses. So in order for the 2 new daughter cells to survive, it needs to have everything. One can't survive without, having ribosomes, without having endoplasmic reticulum, or having all of the necessarily organelles. So once this period goes through, once the cell is finally prepared, it will go into mitosis. So let's take a minute here and to go ahead and see if you can identify and answer these questions. Okay, so now here are their answers. How well did you do? So here's 1 through 3, and then 4 and 5. And so make sure, if you need to, you can pause the video to double check your answers. So now that we have gone through interphase, we're going to go into mitosis. Mitosis is often paired with mitosis and cytokinesis. So mitosis, we're really referring to the nucleus dividing, and hear the word cytoplasm, is the cytoplasm dividing. So let's look at what is happening during this time period. So mitosis, to remember, or just to clarify, mitosis only occurs in eukaryotic cells. So plants, animals, fungus. This will not happen in the same manner that bacteria, because bacteria doesn't have a nucleus. So this is not gonna happen in bacteria cells. Now, it will have a cell division. We call it binary fission, and it's actually a really simple process. But in order for eukaryotic cells to divide, we need to basically disassemble the nucleus and put it back together again. And so that's what we're going to be taking a look at. And then in the process, we also need to separate our cytoplasm and all of the organelles. So remember, at the end of this mitosis and that cytokinesis, we're going to have 2 identical daughter cells. That's what we call them, daughter cells. They're identical, they're twins, they're clones. Each one of them will have 1 nucleus, okay? And so that's what's happening. That's what we're going to be seeing here. We break mitosis down into 4 stages: prophase, metaphase, anaphase, telophase. Um, P, mass, amounts. Today is a way to remember the 4 stages. So let's break these stages down. So in our first phase, prophase, this is when we're looking at a cell. This would be what we normally see as a cell, and it will actually start to break down. And so when it starts to break down, the first thing that we're going to have is the DNA is actually going to start condensing and coiling up into those chromosomes, into those sister chromatids. Up until then, it was that stretched out, elongated DNA cells. This is the point when it's starting to wrap around that histones that we talked about in the chromosomes, and they get really tightly packed. Basically, what we're doing is we're taking all our clothes and stuffing them into a suitcase, right? To go on a trip. Because if they're all over the place, it's going to be really hard to transport those chromosomes around. So that's what's happening here, is our chromosomes are being packaged up so we can easily sort them out and easily move them around. What else is happening here? The nucleolus and the nuclear membrane are dissolving. So they're disappearing. So that membrane that holds the DNA in place is disappearing because we don't want to hold the DNA in place anymore. So we need to get rid of that. The other thing that's happening is spindle fibers are forming in between centrioles. Centrioles are these little almost magnets, if you would, and they're starting to pull themselves to opposite sides of the cells. And in between those centrioles, spindle fibers are appearing. And those spindle fibers actually act as strings to move the DNA strands or the DNA chromosomes around. So all of those things in prophase are occurring. It's a very, very busy time, lots of things going on here. So next, I want to have you look at the cell. So if you can see this cell, these are those spindle fibers we're talking about. You can see these specks starting to form. Those are the condensing chromosomes appearing. Excuse me, I tickle in my throat. So if we were to look at this, and now this is a not your typical microscope. This is a pretty expensive microscope that we're observing this cell under. So now let's take a look at metaphase. Metaphase, my key word here is to remember, is middle. So what we're having here is our chromosomes are lining themselves up in the middle of the equator. So this is, if you remember when you were in like first or second grade and you were having to go to the bathroom and you all had to line up in a row so you guys could all go to the bathroom. Well, that's kind of what's happening here with our chromosomes. Our chromosomes are lining up so we can make sure that we distribute our chromosomes in the correct cell. So each cell gets its correct number. How is this happening? Well, the spindle fibers are attached to the centromere of the chromosome and the sister chromatids, and so they're acting like a puppet to, manipulate those chromosomes to line them up in the middle. And so that's metaphase. And so again, if we look here, notice our spindle fibers, we can see them. Notice at the end of those, the chromosomes. So next we have anaphase, and anaphase are centromere. Remember, the centromere is the tape or the staple that's holding those two identical sister chromatids together that have lined up in the middle. So I've got to imagine a room full of twins, and all of the twins lined up, and one set of twins go this way and one set of twins go that way. So how's that going to happen? Well, first we got to break the staple. So we got to break the centromere. So we break the centromere. The spindle fibers start winding themselves up. They start pulling those chromosomes to opposite sides. And so that's what's happening in anaphase. Anaphase is actually a very short period of time. This does not take very long to happen. As this starts to happen, we are going to start to see the cell membrane will begin to start to cleave and push inward. When we see this, this is what we really think of as the beginning of cytokinesis. So again, if we're looking at it, we can see those spindle fibers, we can see those chromosomes, and we can actually start to see that cleaving occurring. So now telophase. So telophase and cytokinesis, to remember, are happening at the same time. So telophase is the exact opposite of prophase. So whatever we did in prophase, we have to put back together in telophase. So my spindle fibers are breaking down, my nuclear membrane is forming around those chromosomes, my nucleolus is reappearing, and those chromosomes are starting to thin out, and we're starting to see the really thick, dense nucleus that we're used to. So the only time we see chromosomes is during this meiosis stage of the cell. That's the only time we see it. Otherwise, they're all like drawn out and they're all real thin and long. So these chromosomes are becoming thin and long. We're stretching out the DNA during telophase. What else is occurring? Oh, sorry, I forgot about this picture. So here's what telophase would look like. So now what else is happening is we're actually getting the cytoplasm to divide as well. So I'm starting to pinch inward in the cytoplasm. So that cytoplasm is going to start to create a barrier or a boundary between the two cells. And so in plants, they actually form a plate, and a cell wall will actually form between the two. Now, animal cells, we don't have a cell wall, so that doesn't happen. And so we're literally pinching them in and separating into two new daughter cells. Each one of these daughter cells is going to have identical DNA. So if you remember, in humans, I had 72, right, after the DNA replication. So now I'm separating, I'm dividing that up again, so I end up with 46 and 46 again. So let's go ahead and take a minute here to see how you do on these questions. I hope that this clarified what's going on during the cell cycle. Remember, cell cycle, interphase, and mitosis. We have 3 phases in interphase. Remember, we have 4 phases in mitosis. If you have any questions, make sure you're reaching out to me and asking for clarification. Bye, guys.